And continuing on with cell surface signaling, in the previous slide we were discussing protein phosphorylation, but I forgot to mention that as proteins are being transported into the ER, they are glycosylated, and which is a co-translational process. And remember again that this occurs on serine, threonine, and tyrosine residues only of these amino acids, and these ones are the only ones that can be phosphorylated because of their hydroxyl group, which is where the phosphate can attach. But onto this slide, we're talking about cell surface signaling again, and the main point here is to realize that signaling from the cell surface involves cytosolic and nuclear phases, and binding of the ligand initiates a series of steps including a phosphorylation event and a DNA binding event. And we can see clearly that that the kinase phosphorylates the protein, but that in order to do that, there needs to be physical interaction. The first conformational change is due to the binding of the ligand, and the second conformation change would be due to phosphorylation. So now that it's been phosphorylated, it is able to activate processes within the cell. So how does the enzyme become activated? By changing the conformation of the by changing its conformation. And so we should remember these processes, and remember the starting point and the end point, which receptors are able to allow entry into the cell, which ones change. Remember that you do not need phosphorylation in order to make a conformational change. For example, others do it without a conformation, without phosphorylation, for example, glycosylation and methylation. So a little bit more about cell signaling there.